Greetings, my dear friend, my name is Maxim and welcome to my channel. And what I have for you today is something called glycolysis. We will be discussing hexokinase and glucokinase, aerobic and anaerobic as well as pyruvate metabolism, and Warburg effect. Well, without further ado, let's dive into it. The first thing that we have to do is to transfer glucose inside the cell. This action is done by glucose transporters. We will not discuss glucose transporters in this video. After that, glucose encounters hexokinase or glucokinase enzyme in order to be phosphorylated. This step is irreversible and well regulated, so it is worth to remember. These enzymes use ATP molecule to produce glucose 6-phosphate. Basically, if we phosphorylate glucose, it cannot escape the cell because it is polar now. But don't worry, we will discuss hexokinase and glucokinase later in this video. The second transformation is done by phosphoglucose isomerase, which creates fructose 6-phosphate. The best part of this enzyme is that we don't have to remember it. So far we have invested one molecule of ATP, but don't worry, we will gain a profit later. And you will see why. One additional phosphate will be attached, so we end up with fructose 1,6-bisphosphate. This step requires phosphofructokinase 1. This enzyme is activated by high levels of AMP, as well as low levels of ATP and citrate. But the most powerful stimulus is fructose 2,6-bisphosphate. Where this molecule comes from? Well, let me tell you. There is an alternative way where our fructose 6-phosphate by the influence of phosphofructokinase 2 can be transformed into fructose 2,6-bisphosphate. This molecule is very important, as well as its regulation. The main idea here is that phosphofructokinase 2 is a double-action enzyme. It has kinase activity as well as phosphatase activity. There are two major players here who regulates this step: insulin and glucagon. Glucagon acts on its 7-pass receptor and activates G-stimulatory protein, which excites adenylate cyclase, and ATP is converted into cyclic AMP, and protease kinase A phosphorylates phosphofructokinase 2. Importantly, when this enzyme is phosphorylated, its kinase activity is inhibited, but phosphatase activity becomes active. On the other hand, insulin stimulates protein phosphatase, and phosphofructokinase 2 will act as kinase now and produce fructose 2,6-bisphosphate. We have to invest one additional ATP molecule. So far, we spent 2 ATP molecules. And now it's time to gain profit. Aldolase cuts fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. At this very moment, we need help from triose phosphate isomerase in order to convert dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So we will have 2 molecules of GA3P. Our next goal is to create 1,3-bisphosphoglycerate and NADH+ from GA3P. Enzyme GA3P dehydrogenase has 3 domains for inorganic phosphate, NAD, and GA3P. Furthermore, arsenic inhibits this enzyme by competing with inorganic phosphate for the spot. It is important to know that cysteine residue at the active site is essential for this reaction to occur. After that, 3-PG will be produced by phosphoglycerate kinase. What is essential about this is the fact that ADP plus inorganic phosphate will be giving us ATP. This is called substrate level phosphorylation. Our red blood cells have mutase enzyme, which converts 1,3-bisphosphoglycerate into 2,3-diphosphoglycerate. This molecule helps in releasing of oxygen from hemoglobin by cross-linking beta chains. After that, 2,3-DPG can become 3-phosphoglycerate with phosphatase activity in RBC. Whole this cycle is called Rapaport Leubering. And now our 3-PG will become 2-phosphoglycerate with mutase activity. Eventually, 2-phosphoglycerate will be transformed into phosphoenol pyruvate, and the enzyme which is responsible for this action is named enolase. This enzyme is inhibited by fluoride. In the end of glycolysis, we will have 2 molecules of pyruvate and additional 2 ATP from substrate level phosphorylation. Pyruvate kinase is stimulated by fructose 1,6-bisphosphate. In this situation, we can call it feed-forward reaction, and it is inhibited by alanine and protein kinase A. And now I would like to take an opportunity and discuss hexokinase and glucokinase or hexokinase D with you. So, hexokinase is basically inhibited by glucose 6-phosphate, whereas glucokinase is inhibited by fructose 6-phosphate. Furthermore, hexokinase has low Km, high affinity, and low Vmax, whereas glucokinase has high Km, low affinity, and high Vmax. Vmax is a maximum rate possible given the amount of enzyme. Enzymes can become saturated. At Vmax, adding more substrate will not increase the rate. The only way to increase Vmax is by adding enzyme. Vmax can be increased within the context of gene regulation by increasing production of a given enzyme. More enzyme equals more Vmax. Km is a marker of enzyme affinity. Increased affinity will require almost no substrate to start working, and therefore will have decreased Km. Low affinity equals high Km. I want you to think about hexokinase and glucokinase like helicopter and jet respectively. Hexokinase or helicopter has a low capacity for passengers, thus decreased Km. As Km is a concentration of a substrate at which enzyme is saturated by 50%, so our helicopter can become saturated faster than our jet. Therefore, helicopter has decreased Km, but high affinity to the passengers, which means it can start to fly with few passengers on board. Furthermore, helicopters do not fly at high altitude, so Vmax is low. On the opposite side, glucokinase requires high amount of passengers to work, and therefore increased Km. But its affinity is low, and jets can fly at high altitude, thus Vmax is high. There are 2 types of glycolysis: aerobic, which requires oxygen and mitochondria. Its profit 2 molecules of NADH and 4 molecules of ATP. But during the process we lose 2 molecules of ATP. And anaerobic glycolysis, which net ATP is 2. Think like that when you can't make respiration. Think for testes, lactate for lens, when for white blood cells, count for cornea, make for medulla, and respiration for RBCs. Those are tissues which run anaerobic glycolysis normally. This mnemonic was initially created by Dory medicine. Talking about the fate of pyruvate, we have to remember that pyruvate can be transformed into acetaldehyde and ethanol in yeast and microbes. Also, pyruvate can be transformed to alanine, by the enzyme called alanine transferase and vitamin B6, or into the lactate by lactate dehydrogenase, and also vitamin B3 and NADH. Eventually, pyruvate can be transformed to acetyl-CoA via pyruvate dehydrogenase, or oxaloacetate by the enzyme called pyruvate carboxylase and vitamin B7. And last but not least, Warburg effect, which occurs in tumor cells. First of all, an increased rate of ATP production for metabolic needs. Glycolysis produces ATP at a much faster rate than does oxidative phosphorylation. Increased need for glycolytic intermediates for anaerobic pathway required for the tumor cells to grow. Acidification via lactate formation of the tumor extracellular environment, which promotes movement and metastasis of the tumor cells. Increased intracellular signaling through the production of reactive oxygen species and chromatin remodeling via reduction in acetyl-CoA formation, which is a required substrate for histone acetylation. Well, this is all what I have for you today. If you found this video helpful, please leave a like, comment down at the comment section and subscribe to the channel. I wish you a very lovely day and see you guys later.